Welcome to Silverlight Education. In our BZYCT 133 course, we're jumping into Block 3, Unit 13. And wow, are we tackling one of the biggest, most mind-bending questions in all of biology. I mean, really think about this for a second. It all starts with just one cell, a single fertilized egg. So how in the world does that one tiny cell know what to do? How does it build a body with trillions of specialized parts? You know, a heart, a brain, bones, skin, all of it, from the exact same starting point? It's kind of amazing, right? You know, this is truly one of biology's greatest puzzles, and today we're going to unpack the incredible two-part process that gives us the answer. We'll kick things off with the puzzle itself, then watch this cellular explosion called cleavage. We'll see how something like yolk plays a huge strategic role. After that, we'll follow the great migration of cells in gastrulation, and finally, we'll see how it all comes together to build the body's foundational blueprint. It's going to be a wild ride. All right, let's dive right into that puzzle, from zygote to organism. The big question here is, how does a zygote, that single fertilized cell, give rise to an entire super complex organism? Well, the answer isn't some single magic bullet. It's more like a beautifully orchestrated two-act play. And the first act, it's all about creating the cast. I'm talking about a massive, super fast explosion in the sheer number of cells. So that first phase is called cleavage, a cell explosion. And you can really think of it as just that, a cellular explosion. Its whole purpose, its one job, is multiplication. It's all about quantity. So here's what's so wild about cleavage. It's the series of incredibly fast cell divisions, but, and this is the key part, the embryo doesn't get any bigger. The new cells, which we call blastomeres, they just get smaller and smaller and smaller with each division. A great way to picture it is like taking a block of clay and just slicing it up into tons of tiny pieces without ever adding any more clay. So you might be wondering, how is it so incredibly fast? Well, the secret is a brilliant little shortcut in the cell cycle. See, a normal cell, like the one on the right, it has to take time to grow during these G1 and G2 phases. But during cleavage, nope, the cells just skip those growth phases entirely. They just copy their DNA, divide, copy, divide, over and over. This cellular shortcut allows for just an unbelievably rapid multiplication. Okay, but this whole process isn't a one-size-fits-all deal for every animal. There's this one critical factor that completely shapes the strategy, and that's the yolk. So we all know yolk is the embryo's food source, right? It's the fuel for development. But here's the twist. It's also this thick, dense stuff that physically gets in the way of cell division. It's like trying to cut through a really thick, gooey substance. It just slows everything down. So how much yolk there is and where it's located is going to dictate the entire cleavage game plan. And this table just lays it all out perfectly. Check it out. When there's very little yolk, like in an echinoderm, the whole egg can divide completely. We call that holoblastic cleavage. But then you look at something with a ton of yolk, like a bird's egg. Well, the division can only happen in this tiny little disc of cytoplasm right on top. That's meroblastic cleavage. The yolk is literally forcing the embryo to use a completely different strategy. It's amazing. Okay, so cleavage has given us all the raw materials. We have our bricks, so to speak. But, you know, a giant pile of bricks doesn't make a house. You've got to arrange them. And that brings us to Act 2 of our play, gastrulation, the great migration of cells. This is all about organization. This next phase, it involves these huge, sweeping movements of cells, rearranging themselves into distinct layers. But, and this is so important to get, this is not chaos. Not at all. It is a precisely choreographed dance where every single movement has a purpose. Now, we can break this complex dance down into two main types of movement. First up, you've got epiboly. That's where sheets of cells on the outside spread out, kind of like they're stretching to cover the whole embryo. And second, you have emboli, where all the cells that are supposed to form internal stuff move from the surface to the inside. Okay, so how do these cells get inside? Well, one way is called invagination. This is where a whole sheet of cells just folds inward. The absolute best way to picture this is to imagine poking your finger into a soft, partially deflated balloon. See that little pocket you just made? That's exactly like invagination. All right, another way they do it is through involution. With this one, you have an expanding layer of cells that rolls inward over an internal surface. Think of it like a conveyor belt, where one layer is rolling right underneath another. It's this smooth, continuous flow that brings whole sheets of cells to the inside. And last but not least, we've got ingression. Now, this was different. Unlike the other methods where cells move together as a nice connected sheet, here, individual cells actually break away from the surface and migrate into the interior all on their own, like little independent explorers. So, what's the grand finale of all this folding, rolling, and migrating? What's the point? Well, it's nothing less than the creation of the fundamental blueprint for the entire organism. This is where the magic really happens. This blueprint is made of three primary layers, what we call germ layers. First, you have the ectoderm. That's the outer layer. It's going to become our skin and our entire nervous system. Then in the middle, you have the mesoderm. This forms our muscles, our skeleton, our circulatory system, all that good stuff. And finally, the endoderm is the innermost layer. It creates the lining of our digestive tract and organs like the liver and pancreas. I mean, think about it. Every single part of your body can be traced back to one of these three layers. It's incredible. So let's just wrap this all up nice and neat. The puzzle of how one cell becomes a body is solved in two master strokes. Cleavage provides the quantity, the raw materials, the bricks, and gastrulation provides the organization, the architectural plan. You absolutely need both. One without the other just doesn't work. And that, of course, brings us to a brand new, fascinating question. The blueprint is drawn, the layers are in place, but right now they're still just layers of cells. So what's the next step? What process tells a group of ectoderm cells, hey, you're going to become a brain, or tells a little patch of mesoderm to start forming a beating heart? Well, that is the next and maybe the most magical chapter in this whole story of development. We will meet you in the next lecture.